Okay, here we're going to look at cellular nuclear components. And we see a couple cells here with well-defined nuclei and others that have just their chromosomes because they're going through the process of cell division. Well, here we're going to kind of focus on that well-defined nucleus to start with, because it is the command center of the cell, and it's separated from the cytoplasm by a nuclear envelope. It consists of a double-layer membrane. The nuclear pores permit the exchange between the nucleoplasm, which is the interior, and the cytoplasm, which is located on the exterior of the nucleus. Now, this nucleus contains a semi-fluid nucleoplasm, as I just said, that interior space, and chromatin consists of DNA of genes and proteins. This condenses to form chromosomes. So this is important as cells going through the process of division. These chromosomes are formed during that process. So we have our DNA, our kind of isolated kind of patches and particles that are going to be condensed, because every human cell has about 6 feet of DNA in every cell, and it's ultimately going to be kind of condensed down to these chromosomes that we can recognize as this densely packed DNA. The nucleus itself has a nuclear envelope, and that's that lipid bilayer. It's 2 layers here. It's a membrane surrounding the nucleus. It also has an inner membrane coated with laminin, which is a fibrous protein that helps keep everything organized. That's where the chromatin is physically attached to. We have the outer membrane, which is kind of ties in with the endoplasmic reticulum. We have very important nuclear pores, which regulate the transport of proteins and RNA in and out of the nucleus. They're pores, not holes, because they are a regulated step. Now, the reason why these cells or nucleus have these pores, which are very important, they're large protein complexes, and they do allow for necessary proteins to enter the nucleus from the cytoplasm, because they have special sequences that indicate that they belong in the nucleus. A lot of viruses and other things that want to take over the cell want to enter or find a way to get into the nucleus. So these pores are kind of that regulation step. Now, small molecules such as ions freely pass and diffuse into and out of the nucleus, but those larger ones have to go through the pores, and those are regulated. In a nucleus, we have the small darkened region here, the nucleolus, and that's composed mainly of rRNA, which is ribosomal RNA. There's at least one, sometimes many can be seen depending on what cell you're looking at. This is the site of transcription, the site of rRNA where those ribosomes are synthesized and processed. Looking at a kind of cartoon image here, we have that nucleolus. This holds some of the chromatin and ribosomal subunits, region of intensive ribosomal or rRNA synthesis. That's that darkened region. In that nuclear envelope, that's the surface of the nucleus, bound by 2 phospholipid bilayers. It's a double membrane with pores. And that nucleoplasm, which is that inside, that semi-fluid medium inside the nucleus. Those chromosomes, the DNA of eukaryotes, is divided into linear chromosomes, and they exist as strands of chromatin, except during cell division. Histones, these little blue spheres here, are associated with packaging proteins, and they help the DNA wrap around them so they can help increase the density and compact that DNA without it getting tangled.